You come with me, Joe. Welcome to the latest instalment on our Tuesday lunchtime seminar series. It's a great pleasure to introduce one of our own today, or I think Jane would consider herself one of our own now, Professor Jane Pillow. A little bit of history about Jane. Jane was probably destined to be an academic. Her dad was a professor of mathematics at Uni of Queensland. Mum, a microbiologist, and at one stage Mum was working and had 5 kids under 6 years of age. So that's the stock that our today's speaker comes from. But Jane did her undergrad at UQ, then in medicine, then interned in Townsville before training in paediatrics and haematology down in Melbourne, and then came over to WA to do a PhD. She decided she wanted to study young babies, so she worked on preterm infants. And her work basically for a long time was all about how do you get oxygen into babies whose lungs aren't mature enough, don't have enough surface area to get oxygen in. So when I'm talking to people about what Jane does for a living, I tell them she used to basically save preterm babies at king eggs. Then in 2012, Jane thought change of pace might be due, and she decided to surround herself with basic scientists rather than clinicians. So she moved down here to what was then a PhD, anatomy, physiology, human biology. And one of the basic people that she met, I hope he's online, Pete Mark, did what he's done to so many of us.
Pete's in the audience for those who are online.
Shane hasn't got his spectacles on. Pete introduced Jane to the world of molecular biology and the world of circadian clocks, and that basically sent Jane's whole research off into a new direction, the same as it essentially did for our lab, so there's a lot to thank Pete for. And that's basically what the focus of her life now is the development of circadian rhythms in neonates, especially in preterm babies. I've seen some of the data that Jane's going to talk about today, but I haven't seen all of it, and so I really look forward to hearing about what Jane's been up to and especially where the CIRCA DIEM trial is going. Thank you, Jane.
Thanks very much, Shane.
It's great. Oh, get untangled.
Awesome, thank you. Okay, so actually, contrary to what Shane just said, this talk is almost devoid of data. And one of the reasons for that is that most of it belongs to my PhD students and they haven't had a chance yet to publish it or to present most of it. So I'm not going to actually present very much of the data that we've been collecting on circadian rhythm. Circadian rhythm has certainly become a really important component of what we're doing, and I anticipate it's going to continue to do so. We're still obviously doing quite a lot of other research in the preterm sphere down at the PICRU, but the focus of the clinical research has certainly turned away from lung function measurements to circadian rhythms, which are much sexier and much easier to get some funding for. But as Shane said, and Shane, thanks very much for that introduction. I'm not quite sure where the haematology came from. I didn't obviously proofread that bit of the introduction very well. I did think about doing oncology at one point, but it never quite happened. I think the professor of oncology was interviewing me and asked me what I planned to do, and I gave him a big long outline, and then I, being a little bit rebellious at the time, told him I also planned to have a family, and he promptly told me that I couldn't do oncology because oncologists had to be full-time and childless if they were female. So that also changed my life. But anyway, that's not what this talk is about. CIRCA DIEM. So this is really just to try and update the unit a little bit about, not the unit, let's see, I've got to get out of the hospital. Been down here for 10 years and I still forget. Update the school a little bit about the CIRCA DIEM trial in particular and where it's going. But I would like to just start off by, as Shane said, really bringing the focus back to Pete, because it was actually coming to one of these lectures 8 years ago that really changed whole life around. Pete was talking about circadian clocks in the placenta, and I really thought that was quite bizarre. But as part of it, sort of raised some of the issues that maybe were quite relevant to preterm babies, and he really opened my eyes to what circadian rhythms were all about, because up until that point I really thought it was all about jet lag, and some changes in melatonin. That was about the extent of what I understood about circadian rhythms. But what some of you don't realise is that Pete has a remarkable likeness to another famous person. And some of you may recognise this person. Some of you in the audience have got no idea who this is, do you? So this is the $6 million man, Steve Austin. He's the bionic man that we all watched probably before you were born. But that's what Pete is now. Actually, he doesn't realise it maybe, but if you count up my fellowship and our clinical trial grant and the various other grants that we've got to support this work, we've now brought in $6 million to fund this circadian rhythm work in premature babies. So Pete has got a new mantra now as the $6 million man of APHB, but there you go. All right, so things that I learned from Pete. One clock to rule them all, and this is the master clock, is the suprachiasmatic nucleus in the anterior hypothalamus, and it's entrained by Zeitgebers. Most of you will know this, but many of this, some of you may still be a bit unfamiliar. So environmental cues and light of those is most important, but there are a number of others, including food and temperature. And these induce changes in the molecular components. So now, Pete, you're going to find out if I actually have learned anything from you. So you're going to have to correct me if I get this wrong. But so we've got quite a complex clock molecular machinery, and it's all controlled, set in the SCN. So BMAL and clock are transcribed and become the proteins in the cytoplasm, and then they heterodimerize, move back into the nucleus, where they then promote the transcription of parent cry, go out into the cytoplasm, and they then heterodimerize back into the nucleus to inhibit that BMAL clock complex. And then there are some separate accessory loops going on with BMAL promoting the transcription of reverb alpha, which then negatively inhibits BMAL1 expression, and a second one on a separate timeline, which is promoting the transcription of ROR alpha, and that then sets the whole cycle off again. And there are much more complex parts than that, because the core clock genes, you have those clock control genes, and then on top of that, multiple other genes that are under circadian regulation in the peripheral circuitry. So the core clock genes that are oscillating, clock and BMAL and parent cry in opposition to each other, are those clock control genes, as well as a number of others there. E4, BP4 is there, but one is an important one for respiratory circadian function, and then all the target genes that are controlled in the periphery under that. So the other really amazing thing that Pete taught me in that lecture is the sheer number of genes in our body that are under the control of circadian function. So if you look at that big pink circle, so that big black circle is all the genes, just imagine that's all the genes in your body. The pink circle is talking about all the circadian genes, and then you've got genes associated with disease, and you'll see there's quite a lot of overlap with circadian genes, so you can then understand how circadian disruption leads to long-term problems with cardiovascular disease, metabolic syndrome, diabetes, etc. But also there are, interestingly, a number of genes that are specific drug targets that are also expressed in the circadian manner, and so that's now come into chronopharmacology, and the timing of when we get our medicines, that really makes a difference. I don't know, how many of you got your vaccinations in the morning? Yeah, so you probably had a good response compared to those of us who didn't get our vaccinations in the morning to start off with, and got them last thing after finishing work in the afternoon. And that probably, its response to vaccination is also much better if you get vaccinations and you get your immunological challenges early in the morning rather than late at night. So let's tie it back then to the fetus, because the fetal circadian rhythm initially is under control of the maternal circadian rhythm. So the feto-maternal circadian rhythm, really, really vital for growth and development of the fetus. And the fetus is being essentially entrained by all those maternal signals, so sleep, wake cycle, hormones, blood pressure, temperature, body, heart rate, etc. And then the fetal suprachiasmatic nucleus is actually developing relatively late in gestation. So it's critically dependent on those maternal signals for a large part of in utero development. So what happens when you're born preterm? Then you're cut off from that and then deliver and have other things happening to you. But before you're born, there are a number of things that could influence how your body is entraining those circadian rhythms and the nature or the response of your circadian genes. So maternal shift work is known to have some effect on pregnancy outcomes. Ascending infections, chorioamnionitis is also seems to be an important regulator of fetal circadian rhythms, and exposure to hypoxia, as in placental malfunction. This is a big placental infarct there, and that reduces oxygen delivery to the fetus. So just as an example of what's happening with chorioamnionitis, and this is work that Claudia produced last year as part of her honours thesis, so it's one of the few data slides from my group on circadian rhythm I'm going to produce to you today, just because the others haven't had a chance to produce their data or present it yet. I don't want to steal their thunder, but Claudia has presented this and also came away with a prize at Perinatal Society Australia New Zealand earlier this year for her work last year. And this was done in sheep. We implanted biotelemetry sensors into the fetuses and exposed them to saline or LPS, and we were also looking at IDR 1018, which I won't show. And Claudia's work showed that the fetus has got circadian rhythms in temperature and heart rate, blood pressure, respiratory rate that develop and progress over time. And if you look at the left-hand side of this, at the moment, see if I can get the pointer up for those online. So if this left-hand group here, the saline group, so this is our control group, you can see that their circadian rhythms, or the amplitude of their circadian rhythms, gradually increase over time. But if you look at what's happening with the LPS saline group, and this was, just somebody's in the lobby, sorry. All right, so the LPS saline group, when they're given LPS, they initially have this more exaggerated response in their temperature, so bigger amplitude. So that probably just means they were getting a febrile response and then getting a hypothermic response possibly. But then their amplitude of the circadian rhythm in temperature, and also in most of the other parameters that she looked at, decreases over time, so that 7 days later they've almost got complete ablation of their circadian rhythm, the complete opposite to the control group. And this confirms some preliminary work that we actually did, Pete, 2015 with Renita Whittle, with some LPS lambs postnatally. So this clearly, you know, chorioamnionitis, it is associated with up to about 80–90% of preterm births in the extreme preterm age group. And you can imagine already those babies are being born with significant disadvantage for developing circadian rhythms if their circadian rhythms have been ablated by chorioamnionitis. So there are important antenatal factors that may influence development of circadian rhythms, but there are some other implications of this antenatal exposure to infection in terms of what happens to the immune cells in the body, the infection fighting cells, and subsequent infection and inflammation susceptibility. I'm going to come back to that later on in the talk. After a premature baby is born, it comes into the neonatal nursery because it can't survive on its own. So many of them require mechanical ventilation, but certainly require intensive care support over a prolonged period of time. And it's a really abnormal environment. They're either surrounded by bright lights or continuous darkness. There's noises and buzzes and signals going off all the time, so continuous noise, and also continuous stimulation. On top of that, they'll get exposure to both hypoxia and hyperoxia, And on the bottom, that's staph epidermidis, which is one of the most common infections that we get in the neonatal unit. But infants, preterm infants, are particularly susceptible to getting infections, bacterial infections in the unit, and that can have a very detrimental effect on their long-term outcomes. So what's known about delayed circadian rhythm development in preterm, we know that they have reduced feed tolerance. It's associated with some poor weight gains, impaired immunity, increased sepsis susceptibility, prolonged hospitalisation. Long term, it seems that it's likely to be associated with fetal programming for adult onset metabolic diseases. So that's heart disease, diabetes, metabolic syndrome, probably also neuropsychiatric disease in later life. But we were particularly interested to know what's happening for the infants. And when you think about it, what's happening for preterm infants after they're born up until when they were supposed to be born, because this is the time when they're supposed to be in utero under the control of their mum's circadian rhythms and under that really controlled environment. And there are some incredibly important neurological processes happening during that time. So neuromigration, apoptosis, the pruning, refining of all those neural networks, synaptogenesis, all the forming of the connections, and myelination to improve the transmission of nerve signals and coordination post-term. So if the baby is having all of those processes occurring in a setting, an environmental setting where there is no cycling of light during day and night, what's happening to those developmental processes because they're so influenced by circadian rhythms? So the evidence at the moment suggests that it's associated with reduced growth and organ development, so impaired organ development, that you get impaired neurogenesis, you get impaired neurodevelopment, and there's also associations with disrupted circadian rhythms, reduced parental sleep. Actually, any parent of a new child will tell you that they don't get any sleep, but if you've got a child who's got no sleeping at all or no sleep pattern, it's exaggerated. And then that can feed back negatively on the child because it interferes with that attachment between the mother and infant. And we know that that attachment disorder can also negatively impede neurodevelopment. So it becomes a very, very vicious cycle. And as a result, we're really interested to know whether we could actually try and interrupt that cycle long term. So our focus then really became on lots of work had been done on short term outcomes. There was some animal work on the neurological consequences, but there really hadn't been very much human work on what were the long-term neurodevelopmental outcomes of circadian disruption in preterm infants. And we were interested in cognition because that's what parents want to know most. Once their kids actually made it out of the intensive care unit, their next big question is my child going to be intellectually impaired? But there are other things going on neurologically with premature infants, and what many of you may not know is that they have an increased incidence of autism spectrum disorder, ASD, and the incidence is somewhere between 4 to 7 times what it is in the term babies. Now there has to be a reason for that. Now some of that may relate back to, for example, infection in the womb, chorioamnionitis, which may disrupt some of the signalling in the brain. But this was some sort of complicated mapping we put together for an MRFF application last year. Unfortunately, it wasn't successful. But trying to link between the preterm birth and circadian rhythm disruption and the subsequent development of core symptoms for ASD. And this is a really exciting hypothesis that we're proposing to examine further as we go into our trial. So natural history of circadian rhythm development after term, what is known is that generally babies develop their circadian rhythm about 6 to 12 weeks after discharge home. And that's the same whether you're born preterm or born term. The difference is that preterm infants go home a little bit before term expected age. And so you can see that there with this next graph, so this is post-conceptual age on exposure to the home environment at the bottom, and the y-axis there is the time spent in that home environment before babies get a really fully entrained circadian rhythm. And it's the same, 6 to 12 weeks or so thereabouts, regardless of when maturationally you're discharged home. So it's that exposure to that day and night environment that's really critical. So well, what's the big deal? Well, the big deal is the fact that preterm infants are actually in hospital for a long time before they go home. And the more immature they are, the more time they spend in hospital than their more mature peers. So for a preterm infant less than 28 weeks, if you add at least 8 to 10 weeks of hospitalisation onto that, it can be 5 months or longer. And for our 24-weekers, maybe 6 months before they're fully developing their circadian rhythm. And Tash Sorensen, I'm hoping at some point will be able to present to you her PhD findings, as with Carolyn Wilson. They're both close to the end of their PhDs. But they have shown, Tash has shown similarly that our babies, this is data from 1991, so 30 years ago, but this is still the case now with babies in the neonatal unit at King Edward, just in their constant light environment. And that's despite the fact that the American Academy of Pediatrics suggested that cycled light would be a really good way to look after babies about 20 years ago. But nonetheless, most neonatal units in the world have moved from from continuous light, bright light now, more to a continuous dark environment. But it seems that continuous dark is probably still inferior to cycled light. I'm going to show you that in a minute. But one of the main questions here then is, well, are babies, preterm babies, actually capable of having an early restoration of circadian rhythm? If they don't normally develop their suprachiasmatic nucleus rhythmicity of their own autonomous rhythm until late in gestation, is it possible for them to be born prematurely and to have an immediate response? And this is some work from Vasquez Ruiz et al. They did a single centre trial in 2014 that looked at melatonin outcome. Tash and Carolyn have got some really, really fantastic data for this now, and again, I'm hoping they'll show it to you soon. But what you can see is a control group on the left. You've got light bars and then hashed bars there, and there's no difference between night and day over the first 3 weeks of life. But in the group that got hooded, so light shielding over their eyes during delivery, within 5 days of starting that intervention, they had nocturnal melatonin levels that were higher than the daytime levels. So that's pretty impressive. So that's quite a prompt response in a preterm infant to decent light cycling. So what does Cochrane review? Cochrane, for those who have done any clinical research, is kind of the mantra of evidence-based medicines. The inevitable conclusion is that more studies need to be done, and that certainly applies to this. We've got, this is the versus continuous bright light. There've only been 3 studies. They're very, very small, and the outcomes are not consistently done between the 3 studies. Nonetheless, there are suggestions that continuous bright light is negative compared to cycled light, and cycled light reduces hospital stay by over 2.5 weeks and reduces days on ventilation by 2.5 weeks. Wow, that's huge. But these studies were really poorly done, right? Small studies, no randomization, single center, really poorly designed, and who knows what that means. Similar story for versus near darkness. Again, we're coming out with an effect of reducing length of stay just under 2 weeks. But there's some other features that were coming out from this, and that is increased day-night activity. So the intervention cycled light babies were active during the day, sleeping better at night. They had more movements per day in the month after discharge, less crying 3 months after discharge, so they were more settled. But importantly, there was a lot of concern that cycled light and exposure to 600 lux during the day would harm their eye development, but they actually had almost half the incidence, the risk for developing eye disease, retinopathy, or prematurity. So that's very reassuring that the bright light is not harming them. Why have we still not changed? Well, again, these are very poorly conducted studies, very few of them, very low numbers, and there really has been a very strong push, particularly from nursing staff, to developmental care in what they consider to be the darkened environment of the uterus, to try and simulate that. But that mantra has, of course, completely ignored the fact that maternal circadian rhythms are entraining the fetus in utero, and we need some way to simulate that. Noise is much harder. There is some evidence to suggest that just putting earplugs in babies improves their neurodevelopmental outcomes, but again, you can see these are on very few numbers, 12, 20 odd participants, not enough to make any kind of conclusions. But there is another study that had 2 units. This is back in 1986, mind you. One unit went to cycled light, one unit didn't, so it was cycled light and cycled noise. So they had ear protection at night time as well. And the main difference is that the group that had cycled light and cycled noise had fewer hours spent awake, and then also less time spent feeding. But again, this is one centre in each allocation, not really a cluster randomised trial, and nothing that you can hang your hat on. So the sum total of this is that we really don't have very good evidence to support cycled light, although everything seems to be pointing in the right direction in terms of what we should be doing. So that's where we came to the circadian rhythm study, or the CIRCA-DIEM study. And CIRCA-DIEM, most clinical trials now have to have this acronym, and so I was absolutely set on CIRCA-DIEM. I can't tell you how long it took me to come up with that horrendously long title for our study in order to fit circadian rhythm in it, or CIRCA-DIEM in there, but we got there. So cognitive improvement through restoration of circadian rhythms in very preterm infants through environmental modification, the CIRCA-DIEM study. All right, so this was, it's been designed as a multi-centre, so completely different to all the other studies so far, 2-arm, and then what we call a probe trial. It's prospective, randomised, open, because you can't blind the intervention, but with a blinded endpoint control trial. Okay, so it's registered on the Australian Clinical Trials Registry, and the PICOT question, which is how you frame a randomised control trial, population, so, and the intervention, the control, the outcome, or the comparator, the outcome, and the time. So infants less than 32 weeks, the cycled light and noise commenced within 72 hours after birth, continued until discharge home, compared to non-cycled light, so continuous light or continuous dim, and noise, increased cognitive scores at 2 years post-natal age, corrected post-natal age. That's our research question. And that also tells you what our primary outcome is. So we had a whole bunch of secondary aims. We wanted to look at the rest of neurodevelopment, neonatal morbidity, growth and photometry. Wanted to understand a bit about the mechanisms behind, so the biology, the physiology, how babies were behaving, their sleep activity, how they were utilising health service, and also mental health of their primary caregiver. So we only, just had a little study planned, but it's kind of grown hugely from there. All right, so this is the intervention, and parents love this trial. Most nurses really love this trial. It is so simple. It's an eye mask and earplugs, and it's put on from 8 p.m. at night until 6 a.m. in the morning. At night time, we leave their incubator cover on, so that's extra darkness. Daytime, we take that off, or the eye mask, the earplugs come off, and then we make sure that the baby's getting 300 to 600 lux. Why that? Well, we know that you need at least 200 lux to stimulate the suprachiasmatic nucleus into that cyclic pattern, or entrained to light. And 300 to 600 is a reasonably safe level for the eye development. Right, so it's not the really bright light, which is into the thousands of lux that you would be in bright outdoor sunshine. So the comparator for this study is just our control group, and that was just supposed to be pragmatic, so normal unit protocol. The one proviso is that the units need to have less than 100 lux difference between day and night. And that's how we get some separation between our intervention and our control groups. So, and we said constant dim or constant bright because both are still practiced in neonatal units around the world today, although the majority have moved to a constant dim environment because that is in alignment with NIDCAP protocol, which is the neonatal developmental care approach. The one thing that we have changed is that we've stipulated all babies in the trial get their morning coffee in the morning. So some of you may not know, but caffeine is a very regular medication because premature babies get apnea of prematurity. They've got an immature respiratory centre, they forget to breathe, or they have bradycardias at regular intervals, and caffeine is the absolutely perfect drug to get this going, to keep them breathing, which is one of the things you tend to need to do if you want to live after born. So up until we started this trial in my unit, well, I've been trying for probably about 10 years to get them to change to giving caffeine in the morning. What was happening was that babies were getting admitted, they're getting caffeine written up as they're admitted. So if they're admitted at 2 o'clock in the morning or 8 o'clock at night or 4 o'clock in the afternoon, that's when their caffeine was written up, and they got it every 24 hours thereafter. So the one thing that we've said for this trial is all infants will get their caffeine in the morning so that it's not confounding, right? It's not, particularly for our intervention babies, they're not having their sleep at night interfered with waking up in the middle of the night because they're getting a bit of a caffeine hit. So that's been one of the main things there. All right, so randomisation within 72 hours of birth, we've actually just had an amendment to the trial to allow up to 7 days for babies who are in the less than 28-week group. And that's because the parents are often just not quite ready to commit to a trial so soon after birth of an extremely preterm baby, and it's giving them a little bit more time to make that decision. Those babies are in hospital for at least 8 and usually closer to 12 to 16 weeks. And so we decided an extra 3 to 4 days was not critical for that group. We've kept it at less than 72 hours for the babies who are over 28 weeks because they might only have sort of 4 to 6 weeks in hospital. So we're stratified by centre, so every centre will be looked at individually, and that means that individual variances in care will be taken into account, and that gestational age grouping. And the gestational age grouping is really because there's some suggestion in the literature that babies can't respond to a circadian, a cycle light intervention, until after 28 weeks. So we're separately going to be able to look at the extreme preterm, which is less than 28 weeks, to the very preterm. So what we're looking for is a 4-point difference in cognitive score, minimum change to affect a change in clinical management, but it also can be the difference between semi-skilled and skilled work, or between unskilled and semi-skilled work. So it has some importance long-term. The sample size for our study is 954, so that's often 10 times larger, almost all of the trials done to date, and 5 times larger than the biggest trial done to date, which was 165. We're actually at the moment just over 260 babies, so about a quarter of the way through at the moment. So the sample size was adjusted for 10% loss to follow-up, but also we allowed for up to 30% of multiple births because we are randomising the first baby of a twin or a triplet, and then allocating subsequent babies to the same intervention. And that's so we don't send one baby home sleeping well and the other ones not, and trying to understand what's happening with maternal stress and anxiety and sleep patterns with babies on different sleep cycles. So that recruited to date needs to be updated. Sorry, I forgot to update that slide. So this is essentially what happens. A whole bunch of that early stuff here is all just about consenting. We randomise them, they then get their intervention or continued care. We've got prospective collection. There's a bunch of sub-studies that are happening here, and this is what Carolyn and Tash have been doing. And then they're discharged home. Sometimes they're discharged to a secondary hospital first, where we have to keep that intervention going, and then they're discharged home. So I have to tell you that I completely underestimated the ethics and governance involved in this, because when you've got a primary site and 3 to 4 secondary sites for each of those, and you've got 15 primary sites, and every time you put through an ethics amendment, you have to update all of the documents which are site-specific for each one of those sites. We end up updating somewhere in the order of 200 documents with each ethics amendment, and it is just horrendous. So we are just about to employ another person almost entirely to just keep on top of our governance arrangements. So it's no small matter to try and manage this through the clinical trial process. Once they go home, they're then getting a whole bunch of interim follow-ups. So running the whole trial on REDCap, which is just an online thing, but the follow-ups, lots of questionnaires that people can receive on their mobile phones, smartphones, and just type in the answers, and then it goes back, automatically goes into our database. So the follow-ups are looking at infant development over time, their growth and development, mum's mental health checks, infant movement, so parents are taking videos of their infants, uploading them, and then they're getting assessed for general movements to assess whether they're at risk of cerebral palsy or not. And then there's some child behaviour checklists around 22 to 23 months, and then they finally come back for a check at 2 years, corrected postnatal age, and that's when they're getting Bailey 4, which is our primary neurodevelopmental follow-up assessment. It's got sort of language, speech, motor, cognitive components to it, and they'll get their anthropometry. Mum will get their final mental health check there. Babies who are screened on the behaviour checklist of being at risk of a social communication disorder are then going to have an extra assessment specifically looking at autism. So there's an autism sub-study built into this. All right. So we have our core study, and as I mentioned, there's been a bunch of different sub-studies that are arising out of this. This is becoming bigger than Ben-Hur, and it's about to become even bigger because we're adding a whole new area down here, which is respiratory infection, which has again come out of just a paper I happened to come across at the end of last year on susceptibility to infection with circadian disruption. So I wanted to spend a little bit of time talking a bit about that because ultimately my origins are in the lung. And so I thought it'd be nice to round the talk out talking about that briefly. And lung development is very closely linked to the expression of the circadian system. So on this graph here, this is the PAIR1 gene, which you'll remember is one of the core clock genes that you have. And looking at its expression in various tissues in the lung, the endothelium, epithelium, mesenchyme, and the alveolar parenchyma, right from the postnatal age through to adulthood. And that you can see that its expression changes over time, but it's really important during those early developmental years when the lung is still actively growing. So there are increasingly awareness of all the signals and factors that can influence lung development, and if they go wrong, then result in chronic airway disease. So with airway hyperactivity, susceptibility to infection, remodeling inflammation, altered contractility. And there are things that can happen and influence that, the inflammatory response and antioxidant defense, that are also influenced by clock genes and circadian rhythms. So the proposal here is that circadian disruption can have some impact to actually promote chronic airway disease and disorders throughout childhood and later life. And that's further emphasised here when we look down at what's happening at the molecular phase, and you'll recognise the genes in blue, so many of the core clock genes. The genes in yellow there, so this is the Wnt and the BMP, the H1F targets, for example, they're all developmentally really important genes in the lung, and they're influenced by core clock genes. The red genes are not particularly influenced by any circadian expression, but these green genes that influence the inflammatory responses are modulated by circadian rhythm, as are various other intermediaries like CAMP. So it's quite likely that circadian disruption in early life could impact on subsequent growth and development. And antenatally, that seems to be likely also the case in terms of when the clock genes are expressed right throughout development, and it will depend on the cell specificity and their differentiation rate. But essentially, this may involve some fetal priming for postnatal life. And if you are born prematurely and have very delayed development of your circadian rhythm, then potentially you've got some kind of program disruption that will program you towards that chronic airway disease. So why is this relevant, or why are we paying attention to this? So this is some work from my colleague Shannon Simpson up at Telethon Kids on various preterm cohorts that we've had from King Edward that have then gone on to be followed up with the respiratory team up at Telethon Kids over the last 20 odd years. And one of the things that she has been able to show is the steady decline in lung function throughout childhood that's occurring, not only in those kids with bronchopulmonary dysplasia, it's not come up there, but sorry, the BPD is the, yeah, is my legend here. So BPD are these really dark constant lines here, and these are preterm infants dashed lines without BPD. So regardless, both groups are having a decline in lung function. Obviously, those with BPD are having a more exaggerated decline. But why are kids without BPD also having decline in their lung function throughout childhood, whereas babies born at term who are healthy don't? Right? So something is fundamentally happening and different to make these kids have a dysregulated lung function that progresses through childhood. And one of the questions that we're asking now is this a consequence of dysregulated response to inflammation? So are they getting some kind of inflammatory lung disease as a result of their responses to childhood infections, airway irritants that are actually causing that? So this is a really interesting little GIF that I found, and this is looking at the wounds that are happening during the active phase of the cycle, of the circadian cycle, you see is in the left GIF, versus wound healing on the right that's occurring in the quiescent phase of the circadian cycle. And there's a fundamental difference between those two in terms of the rapidity of wound healing. So circadian function is, or circadian rhythmicity, is a really integral part of our response to injury and infection and how we heal and recover from that. And if you can think of viruses entering the respiratory tract, and what many of us are feeling, I know I'm coughing almost continuously, usually I'm waiting for the spasm to happen right now, but you've obviously got a constant airway inflammation that's going on, and it's how rapidly do you heal from that. So if you've got a disrupted circadian rhythm, perhaps you've got this exaggerated or prolonged inflammation that may actually contribute to ongoing lung function disruption. So this is another paper that I came across last year that was really quite interesting. This is a group in the US, in Philadelphia, who have taken mice. They took actually adult and neonatal mice. I'm only showing you the neonatal ones here. And I'm sorry that those graphs haven't exploded up very well. But basically the neonatal mice, they took them and they gave them a 5-day exposure to either hyperoxia, 95% oxygen, or to room air, and then let them recover and grow out to be adults. And then they exposed them as adults to influenza A virus. And they exposed them at ZT11, cyclibutan 11, or at ZT23. So 12 hours apart, 2 different times of the day. And you have a natural increased susceptibility to infection during the quiescent period. So those in room air, you can see the difference between the ZT23, sorry, let me just show you here. So this was the ZT23, so that's during their active phase, and then ZT11 is this red dot here. So there's a difference night and day in terms of susceptibility to infection. But those that were exposed to hyperoxia lost that time of day susceptibility. They just were absolutely increased susceptibility to infection right throughout the day. And what they subsequently found is that that was entirely specific to the loss of BMAL1 in the airway type 2 epithelial cells. So they did a knockout of the BMAL1 and were able to demonstrate exactly the same susceptibility, loss of susceptibility, time of day susceptibility to infection. So the mice were throughout. So it suggests, and subsequently they also found that that was actually an epigenetic phenomenon in that. So there's something about that initial exposure that is having a long-term effect on the expression of BMAL1 and other core clock genes that is changing lifelong susceptibility. And that didn't happen when adult mice got the same exposures. So thinking about that and inflammation, and then just thinking about everything that happens in an intensive care unit, there is a huge amount of the physiology of ICU patients that is fundamentally related to infection and inflammation, and the cytokine responses, the capillary leak syndrome, wound healing, responses to infection, etc., and also just the energetics of cell function. And all of those are influenced by circadian functions. So if you get a disruption in your circadian expression or just machinery, then you've got potential for long-term damage resulting from that. And that's shown here in, so this is another sort of version of the mouse experiment. If you look at innate immunity against pediatric viruses, so this is BMAL1, and they've got some knockouts in some mouse embryonic fibroblasts, the MEF, but then they also did it in an adult cancer line, which was the A549, where they did a knockdown of BMAL1. And they were able to demonstrate that loss of that BMAL1 resulted in higher viral titers that were really persisting in the lung for much longer in the absence of proper BMAL1 function compared to the wild type controls. So we might have not just increased susceptibility to infection, but more prolonged infection that may sustain that inflammatory response and then contribute to long-term dysregulation of lung function. And of course, there are other environmental agents, and cigarette smokers is probably key amongst them in our preterm population, because prematurity is much more common in our lower socioeconomic classes that tend to have more smoke exposure, and passive smoke is a big problem. Exposure is a big problem for our premature infants. So those things may also contribute to dysregulated lung function. So where does that all take us? Basically, we are now just starting another big part of the CIRCA DIEM trial, a new bit that's in development and probably will go to parent later this year, to look and see if we can understand the mechanisms behind respiratory susceptibility in childhood, and whether circadian rhythm will contribute to that. And we've got a perfect population cohort to study this in, because we've got babies that were exposed to a circadian intervention early in childhood. Mini spoiler that it improves their, they get earlier restoration of the circadian rhythm, and infants who don't, who have just got normal exposure and then have that delayed circadian rhythm development. And so we're going to be looking and taking some samples from these babies at baseline, have their intervention again before they go home, and it will be looking at neutrophil function, looking at blood immune profiling, also collecting some airway epithelial cells, which will go for culture, where we've got folks up at the Institute doing a lot of work on responses to wound healing and viral infection in airway epithelial cell cultures at the Institute. So some work that's going on there, and this is just in development at the moment, but then we'll bring them back at 6 months and again at 2 years to do some more functional studies on their immune function, neutrophil function, but also ultimately to look at, have they had increased hospitalisation over the first 2 years for respiratory tract infection or other infections of any sort. So these are the folk involved in this is Luke Garrett, Tom Isophides, Anthony Kisix, Dave Martino, Shannon Simpson, Pete, of course, because he pops up everywhere, and Mark Nicol, who's from microbiology and is going to do some viral work for us on that. So just wrapping it up, it's been a pretty long journey. We started, actually it goes before this, doesn't it, Pete? Because it was 2015, I think, that Renita did her first work with us in PICRU. But we have had 3 NH&MRC applications. We were finally successful in the middle of last year. We've had various other small bits of grant funding along the way. Telethon, we're now looking for some more funding for the respiratory sub-study, but it has not been for the faint-hearted, but it's now, I'm really proud of this trial because it's not only just going to be looking at the clinical outcomes, but it has really incorporated within it a whole bunch of basic science to understand mechanisms behind all of this so that we're actually going to be able to explain what we find and have some implications long term. So I'm anticipating there's a growing awareness and interest in this around the world. We're quite keen now to try and develop a perinatal circadian rhythm network around the world. I'm keen that that's coordinated from Perth and as part of a push within the school, possibly towards a centre of excellence in circadian biology at the university level. Like most things, it doesn't happen without a lot of help, and I want a really huge shout out to a couple of people. Pete's already had a $6 million moment, so not going to give any more kudos there, but Tash Sorensen and Carolyn Wilson, who are the 2 PhD students, without whom this trial would never have got as far as NH&MRC funding, because they just did a huge amount of work on this trial in the early stages, and it's really been pivotal to us getting that funding. And then all my other chief investigator collaborators, Rod Hunt from Monash, Andrew from up the road, thank you, White House, thank you. Yeah. Julie Marsh, who's our biostatistician, Alicia Spittle, who's cerebral palsy, Peter Anderson, who's neurodevelopmental outcomes, Nadia Badawi, again, cerebral palsy and clinician, and probably heaps of associate investigators also that I have not included here, but huge cast of thousands. So there are a few take-home messages there, but that, you know, fundamentally, I hope I've given you a bit of an update. I've gone a little bit over time, I'm afraid, not left much time for questions, but I think this is a fantastic study and is really going to do a lot to bring circadian biology in premature infants to the fore. So thanks to all our funders, and left you 5 minutes for questions, so sorry.
Thanks, Joan, for a great journey. Somebody's got to get a camera out. Pete definitely needs a new photo. You must have collaborators come down and say, is your son around?
You can just use the Lee Majors one if you want.
So, questions for Joan?
In the big study, when the infant is sent back home, do you give any advice to the parents on how they should actually manage the day and night cycle?
The only thing we tell them is that they're not allowed to use an eye mask or earplugs after they go home. And that's primarily because of safety reasons. In hospital, you know, the eye masks are potentially a suffocation risk. The earplugs, if they got out, they could be an aspiration risk. And particularly as babies mature at home and start to get more coordinated with their hands, they
I was
cause damage.
more thinking of people who can leave a light during the night.
Yeah, no, we don't give any information at all. Basically, we've just confined it to being an intervention during the hospital period. But you're right. I mean, we could.
But you could ask them.
We can ask them if they're doing it.
Yeah.
We could, you know, arguably we could have given that advice to the intervention group and not to the control group.
Yeah.
But we figured that just being in the trial, that there was going to be a heightened awareness of circadian physiology in both groups, and they probably were both going to try once they got home to get the babies settled. Yes, Archer.
So fascinating. My question is just a practical one. If the study shows that your intervention is great, what would a gold standard NICU look like? Would it be always light but with masks on, or would it be light, dark, like trying to mimic?
Yeah, so that's a really interesting question. And when Pete and I first started designing this trial, we got really complex and we were trying to give babies melatonin at night and some cortisol in the morning, and that just was not going to go down with my neonatal colleagues at all. But we started talking about a circadian rhythm package. And so, you know, part of that would be develop timed care, so trying to reduce cares overnight as well. There are neonatal bed units now that have this phased light up and phased light down, so that you get a day and a night and a dawn and a dusk. But you know, many units couldn't afford that kind of thing. I think the real attraction of this is that even changing out the eye masks a couple of times a week and the earplugs every few days, it costs us about $60 per infant to do this, and that is against a $2,500 per day or $200,000+ stay in NICU per infant. Is a minimal exposure, and even if you paired that back to just having one pair for the infant for the whole of its duration, it's doable in third world developing countries where they don't have any money to change their NICU environment. So it's incredibly pragmatic and globally applicable intervention.
Can I just add to that? So, I mean, we thought about the melatonin and cortisol in there as well, but that, as Jane said, a step too far for the outcome. But what I'd really be interested in doing is getting their actual cribs, because they don't regulate their own body temperature particularly well, and giving this light, subtle circadian rhythm in body temperature as well on top of, that would mimic what they see in utero.
Yeah,
so
and that will take a step, because our nurses are absolutely entrained to keeping the temperature at 36.5 degrees right throughout the day, and they get very disturbed when the temperature falls overnight just before babies get home, which is probably because they're physiologically behaving.
So what you're saying, Jane, is the temperature of the baby is monitored at all time, and if when you put the mask on, their physiology change during the night, they crank up.
Yep.
That's the next battle.
Yeah, it is the next battle, but it's a step too far for this trial. I think we have to demonstrate the importance of circadian rhythms first, and then we can start promoting a circadian appropriate temperature regulation.
Can I ask a question?
Yep.
Yeah.
Who's that online?
Ingrid.
Yep, go ahead.
Jane, I was looking at your innate immunity data, and we found that a subset of children at high risk of asthma don't produce an efficient interferon immune response to viruses. And it's something that can actually be fairly easily tested when children have a virus. I was wondering if you would consider including that in your study.
In terms of measuring their interferon responses?
Yeah, so you take a nasal swab and you do some fancy analysis of it and determine what their interferon response is during a viral infection.
Ingrid, that's entirely possible. We're certainly looking and considering doing viral swabs during the respiratory sub-study where we see an infection, and probably talking about using Steve's Aerial app to monitor those infections. So that would be certainly doable. And we know in infection also that in asthma there's also circadian variability right in asthmatic responses. So makes sense.
We sorry, I'm trying to get back to the point. We found that's independent of the virus they're infected with. So although in asthma, rhinovirus is the predominant virus, the deficient interferon responses were not related to the viral infection. It was an inherent deficiency.
Yeah, I suspect that that's true, and then exacerbated by viral illness.
Yeah.
I think we're probably going to have to wrap up, aren't we?
Yeah, I think
because we've got somebody.
And there's a class coming in.
There's a class
so
coming in. Sorry.
Please join me in thanking Jane, and I for one look forward